Hi everyone, my name is Tessa Passion. My home university is North Carolina State University. My literature review was on the role of brown adipose tissue, and it was titled Significance of Brown Adipose Tissue with Respect to Non-Shivering Thermogenesis in Newborn Animals and Its Relevance in Humans. So to go through the abstract, my review analyzed the thermogenic ability of brown adipose tissue in newborn animals as well as its role in the human body. Brown adipose tissue is mammalian tissue that protects the newborn animal as well as the human's body temperature at birth. It is responsible for non-shivering thermogenesis of newborn animals through lipolysis. Brown adipose tissue contains numerous mitochondria, multiple small lipid droplets, and uncoupling protein 1. Uncoupling protein 1 is an inner mitochondrial membrane protein that is only present in brown adipose tissue. Its function is to uncouple oxidative phosphorylation from ATP production, causing the dissipation of energy as heat. This dissipation of the energy as heat is critical in maintaining body temperature immediately after birth and also immediately after hibernation in some of the hibernating animals. Brown adipose tissue is made before birth, and it makes up about 2 to 4% of the birth weight of the animal. Newborn animal, however, loses its ability of non-shivering thermogenesis 7 to 10 days after birth. Throughout this literature review, brown adipose tissue development in ruminants will be discussed, as well as the supplementation in sheep in order to increase brown fat deposition. Even further, the role of brown adipose tissue in humans will be explored to better understand its significance. So, a little bit of an introduction. Brown adipose tissue, like I mentioned, is found in newborn animals and is important immediately after birth. The quantity of brown adipose tissue peaks at the time of birth in order for postnatal survival to occur, and it gradually decreases during the early weeks of life. Catecholamine release is the stimulus for brown adipose tissue thermogenesis in the early neonatal period, and responsiveness to catecholamines is markedly increased by the cutting of the umbilical cord. Brown adipose tissue contains large amounts of mitochondria as well as uncoupling protein 1, otherwise known as UCP1. UCP1 allows for the generation of heat, and it is a transport protein that uses fatty acids to facilitate the transport of hydrogen ions across the membrane. UCP1 expression is also positively correlated with metabolic inefficiency being increased by cold acclimation. Throughout this review, I will talk about studies that compared brown adipose tissue between Angus and Brahman calves, supplementation in sheep, brown adipose tissue as an anti-obesity agent, and browning of white adipose tissue. So just to give you a little bit of a background of brown adipose tissue, I attached a picture here. You can see the difference between a white fat cell and a brown fat cell. Here are all the mitochondria I've been talking about and the multiple lipid droplets, and you can see that a white fat cell doesn't have this at all, and the mitochondria is what gives the brown fat cells its brown color. So a little bit of a function of brown adipose tissue. It's, like I said multiple times, extremely important in newborn animals. It contains numerous mitochondria and small lipid droplets, and it varies within breeds of species as well as species to species. Brown adipose tissue also uniquely possesses UCP1, which allows for the generation of heat within these animals. It's responsible for non-shivering thermogenesis, which ultimately increases the survivability of these animals. This is important not only for the animals overall, but the producers as well. Like many of you probably know, the ability of animals to keep themselves warm at night when animals calve or lamb or anything like that, when the producers aren't not paying attention, but when they're, that was a bad analogy, when they are out doing something, or when they're finally able to go to bed, or the few seconds that the producer walks away from the animal, lots of times that's when they decide that they're ready to have their baby. And it's important in those critical moments when it's cold in the winter, because they come out wet and everything. So this really helps with the survivability of these calves, because it helps protect from hypothermia. Like I said, it's important to the producers as well as the animals, because if the animals kept dying, the producers wouldn't be making any money. So moving on to the thermogenesis, here's a picture of just a stained slide of brown adipose tissue compared to white adipose tissue. UCP1 is specific to brown adipose tissue. It allows mitochondria to oxidize substrates rapidly without ADP phosphorylation, thus uncoupling oxidation from phosphorylation and instead promoting the dissipation of oxidation energy as heat. So brown adipose tissue is a specific transport protein of the inner mitochondrial membrane that increases this inner mitochondrial membrane conductance for hydrogen ions to dissipate the mitochondrial hydrogen ion gradient and convert the energy of substrate oxidation into heat. So that just even further explains why all brown adipose tissue has all these mitochondria, and that's how heat is produced. And this heat is what allows the body to maintain temperature without shivering. UCP1 is activated by long-chain fatty acids that are produced within brown adipocytes by the lipolysis of cytoplasmic lipid droplets upon stimulation of brown adipose tissue. Along with non-shivering thermogenesis, BAT is responsible for the cold acclimation-recruited catecholamine-induced thermogenesis. Heat production from brown adipose tissue is activated whenever the animal or human is in need of extra heat. Such instances include immediately, postnatally, during entry into a fever, and during arousal from hibernation. The rate of thermogenesis is centrally controlled via a pathway initiated in the hypothalamus, and it is dependent on the catecholamine release from the sympathetic nerves. So moving on, this was a study done by S.B. Smith, and the purpose of this study was to compare BAT between Angus and Brahman calves. I'm sure many of you are familiar with Brahman and Angus, but this right here is the Brahman, and this right here is the Angus. Brahman are tropical breeds, and the calves exhibit higher or greater cold-induced mortality than regular Angus calves. Smith's study determined not only that BAT or Brahman calves contain 2 to 3 times as many total beta receptors as Angus, but there was also a higher presence of mitochondrial UCP1 in the Brahman brown adipose tissue. Smith collected fetuses that were taken at 96, 48, 24, 14, and 6 days prior to parturition and also at parturition. He found that the lipogenesis from acetate and glucose in vitro decreased 97% during the last 96 days of gestation in both Angus and Brahman calves. He also found that UCP1 gene expression tripled during gestation in both breed types. And then within another experiment conducted by S.B. Smith, he exposed the calves of each breed type to either 48 hours of warm exposure or 48 hours of cold exposure after the initial 15 hours of the regular newborn animal treatment. He found that Brahman BAT adipocytes shrank with cold exposure, while Angus BAT did not shrink with the same cold exposure. Overall, this study, with his other studies that I just previously mentioned, indicate that BAT tissue from Brahman calves may be exhausted of lipids shortly after birth during the exposure to a cold environment. He reported that calves with greater Brahman inheritance exhibited higher morbidity and mortality rates during the neonatal period, especially as ambient temperatures decrease. Moving on from cows, a study done by Budge found that increasing the quantity of feed provided in late gestation in sheep increases fetal weight and also promotes brown adipose tissue maturation, and the combination of which will overall enhance neonate viability. Budge had an experimental group that he fed the pregnant ewes 150% of their dietary requirements, and then he had the control, which met 100% of their dietary requirements. He found that the fetuses sampled from the well-fed, or the 150% diet ewes, were heavier but possessed less brown adipose tissue per kilogram body weight, which in turn had a greater UCP1 abundance and higher thermogenic activity than the control fetuses, which were the ones getting 100% of their dietary requirement. In sheep, brown adipose tissue develops in late gestation and disappears shortly after birth, as I've stated before. And a study by Wu found that supplementation of L-arginine reduces white adipose tissue in obese pregnant sheep. Another study that deals with supplementation by Clark found that algae supplementation a study by Keithley found that supplementation with algae actually had a negative effect on lamb body weight, but in fact increased the specific gravity of the used colostrum. And then a final study by Clark measured the thermogenic activity of lambs fed 50 milliliters of water at 5 hours of age in either a warm or a cool environment. And the major finding of his study is that feeding a small volume of warm water to neonatal lambs can have a significant effect on temperature and plasma concentrations of catecholamines and metabolites that is dependent on the ambient temperature at which the lambs are maintained. He found that feeding a small amount of water can influence thermoregulation that is dependent on, like I said, the temperature that they are maintained. So the results of his study mainly showed that the temperature that they're in, as well as feeding them warm water, can play a major role in determining the need of supplementation in sheep overall, which, like I mentioned before, is a challenge to producers in general. So moving on, and kind of connecting the whole One Health mentality, I found a few studies that dealt with cold activated brown tissue in men. These studies evaluated the presence, distribution, and activity of brown adipose tissue. The study found that in 23 of the 24 subjects, in 23 of the 24 subjects, brown adipose tissue was found under cold monitored conditions. And then in another part of the study, 10 brown adipose tissue was actually lower in the obese subjects compared to the lean subjects. Another study involving people, 51 subjects, both male and female, about 27 showed cold activated brown adipose tissue and 24 did not. Then the study went on to measure capsinoids, and capsinoids, they're known to be food ingredients that increase energy expenditure and decrease body fat. So these people were given the capsinoids, and after 6 weeks, Yoni Shiro, who did the study, found that the energy expenditure as well as a slight reduction in body fat in these test subjects. And then a final study found that brown adipose tissue can possibly prevent obesity. A progenitor cell was found in adipose tissue that can become brown or white depending on the stimulus. And then for the future, just going off that last subject, depending on the stimulus, if that stimulus can actually be determined, that would be huge to help prevent or, have an anti-obesity effect on some people, or the browning of white adipose tissue would be huge. And then on the animal side, genetic selection for animals with high brown adipose tissue could be a great thing, especially for producers that live in areas like, let's say, where I'm from right now, Champaign, Illinois. We got our third round of snow. The past 3 Sundays we've gotten snow, and usually this is when calving season starts, so that genetic selection for animals with high BAT would be very beneficial. And then maternal supplementation as well, that could be a seasonal thing. And finally, I found some information on the fitness industry. This isn't research-based or anything like that, but there are ways that the fitness industry is activating and targeting brown adipose tissue through what you eat and how often you exercise, and exercising in cold environments and taking a cold shower, then eating high energy foods right away and then working out. There's all these different ways that I have found that aren't any factual based information. I just wanted to throw that out there. And then I have just a picture of me on one of my favorite cows. If If you have any questions, I know that I had to talk really fast through this, please email me. And if you found anything with any information that you found interesting, email me as well.